Okay, thanks. So we are going to start a new lesson. The remaining parts, we will get back to it after a while. Today, I would want to finish a chapter on it. It's from the fourth unit. Okay. We're trying to learn some applications. That's the essence of what we're trying to learn here. So it is about strategies towards beta cell replacement. It is mostly about diabetes. I hope you remember um, reading about diabetes before. If not, we will have a good enough discussion in this as a part of this lesson. Diabetes, there are two, I guess you know it, diabetes mellitus and diabetes insipidus. Insipidus is about lack of vasopressin and frequent urination, loss of water, excess water. Whereas diabetes mellitus is about hyperglycemia. Hyperglycemia is having high blood glucose. Okay, this blood glucose, having high blood glucose for a prolonged time can affect many other organs, and that is what we will try to learn in today's class. So we will also try to learn a little bit. There are some papers which are, majority of the experiments are being done to discuss about reversing diabetes within mice. Okay, we are not going to discuss that, but we will see what are the approaches that are possible, and I hope you realize that majority of these are under clinical trials. Okay, there are many types of diabetes, or there are several causes for why there is hyperglycemia, high blood glucose. At least the three predominantly understood ones are type 1 diabetes, type 2, and gestational ones. And while we are mostly concerned about these two, diabetes 1, type 1, and type 2. Type 1 is characterized by lack of beta cells. Beta cells are destroyed mostly because of autoimmune responses or other viral infections. Type 2 diabetes is mostly about resistance to insulin. In the type 1 diabetes, there is a lack of insulin. In type 2 diabetes, even though there is insulin, the response, there is lack of response to insulin. That is resistance to insulin. Okay, these are type 1 and type 2. The gestational one, for somehow we do not know complete details about it, or at least the prevailing one is there is high blood glucose, and because of which especially the tests for if there are any pregnant women around, they do get their blood glucose tested, because when there is high blood glucose, it brings in more glucose to the baby, and the baby might grow excessively, which may cause delivery issues and other things. So the doctor may suggest precautions accordingly. Type 1 diabetes is mostly, as I said before, it is destruction of beta cells. Beta cells are the ones that are producing the insulin, the blue ones indicated here. And we do not know completely why this happens, but there are three major factors probably involved. Are one of it is autoimmune disease, as a result of which the beta cells are killed away. Second thing, some people are highly prone to diabetes because of some mutations or some kind of genetic changes. And there are also other conditions like viral infections, which could cause the lack of beta cells. Again, before going further, we will we will also look at type 2 diabetes, and we'll have a comparison in both of these cases. What does insulin do? Insulin is produced by the pancreatic beta cells, and the insulin is goes to the target cells and will stimulate the cells to take up glucose. Every time there is high glucose in the blood, insulin is secreted by pancreas, and insulin stimulates the target cells like this one to take up glucose, and then blood glucose goes down, right? Whereas in type 2 diabetes, although there is insulin, and although the insulin may bind or not, the point is that the cell is no longer responsive, and therefore glucose is not taken up by it, and as a result the blood glucose increases. That is hyperglycemia or diabetes mellitus. Let's just compare one of it. In type 2 diabetes itself, there are kind of two phases. Let's see the healthy one, where this is healthy one. The pancreas produces insulin, insulin binds to insulin receptor, and glucose is taken up, and the blood glucose comes down. Whereas in type 1 diabetes, we have the beta cells are killed, because of which there is deficiency of insulin. So there is no stimulus for the cell to take up glucose. So the blood glucose keeps high. In type 2 diabetes, what happens is insulin is present, but somehow the cell is not able to be, or the insulin fails to stimulate, or the cell becomes resistant to insulin. So the glucose is not taken up. As a result, the glucose remains high in the blood, and that is hyperglycemia. In type 2 diabetes itself, we can also have two phases. The first is the insulin resistance, and then the insulin deficiency. Insulin resistance is when the target cells do not detect or may not have any effect because of the insulin binding. That is the resistance phase. As a result, so in this case, for example, the blood glucose is high, insulin is produced, still the blood glucose is high, so pancreas keeps producing more and more insulin, and as a result, at one point, the beta cells that overwork, and they may undergo through apoptotic mechanisms, may undergo cell death because of the exhaustion, wear and tear. And in that case, it is also insulin deficiency. First of all, the cells are insulin resistant, and also the second phase is there is no more insulin produced by the beta cells. Okay, so which are these are usually associated with obesity and lifestyle and many other things. Okay, let's look at what cells and what are they producing, and how the glucose homeostasis happens within the body. If you see the pancreas, there are three different or four different types of cells: alpha cells, beta cells, delta, and there are F cells, and also exocrine pancreas. So the beta cells are the ones that produce insulin, and alpha cells produce glucagon, and delta cells produce somatostatin. Right? In type 1 diabetes, these beta cells are killed, because of which there is no insulin production. Let's see what these hormones do and how important pancreas plays in the metabolism of an individual. Beta cells are what we are discussing. It is to increase the glucose uptake by the target cells. Usually the target cells are the skeletal muscle cells and also liver tissue. So, and this will, as a result of which, it will reduce the blood glucose. It can also increase the synthesis of glycogen, that is glycogenesis, right? And it lowers the protein breakdown. Glucagon, on the other hand, it is more of the reverse to that of the insulin. Anytime the glucose levels have fallen to blood glucose levels have fallen beyond a certain point, or there is an excess need for blood glucose to increase to supply to the rest of the body, then also glucagon has a function. Glucagon undertakes glycogenolysis, right? It mediates, these are two. Insulin is favoring glycogenesis, and glucagon is favoring glycogenolysis. It increases the blood glucose levels, right? And these two are the important ones as per what we are discussing at this stage. But otherwise, delta cells, um, they produce somatostatin, which is involved in many other functions. So what we are talking about is high blood glucose, hyperglycemia, and that is affecting so many people all over the world. And even in India too, there are large scale problems associated with diabetes. We are trying to uh, trying to generate motivation, and at the same time understand the biology, and hopefully, and also understand the ways people are trying to use beta cell, how to treat diabetes. The main symptoms of diabetes, I think every family has one or somebody or some friends or distant relatives who are having diabetes. In my home too. So there are some of the symptoms are systemic weight loss and breath smell of acetone, because there are because there is no more glucose uptake, so body uses other types of through gluconeogenesis. It might take up perform other biological metabolic pathways, because of which they may have a smell of acetone when we're breathing. There are nausea, abdominal pain, and these are some things very common, that is polydipsia and polyphagia. That is, you feel hungry, you feel thirsty all the time, you feel weak, and sometimes when it advances, the vision is also blurred. And also, this is another thing, previously they used to test the urine glucose. Polyuria is frequent urination, and glucosuria is present of high amount of glucose in the urine. So diabetes as a symptoms is something, and as a complicated disease, it's like you're hitting something, and because of which many multiple other things are also hit. It affects many things, including it causes diabetic cardiomyopathy, diabetic retinopathy, periodontal disease, diabetic nephropathy, that's the kidney problems, dental problems, even encephalopathy. So diabetes is just not about sorry it's just not about blood glucose, but because of high blood glucose, many other tissues or organs are actually affected, and these are some of the prominent ones that are affected. We will see a bit of each, a little bit, and let's see. This is a normal eye, and diabetic retinopathy may also happen. What this means is we have a layer of the retina inside, and there is blood supply, and sometimes some of them may start breaking down, or there might be hemorrhage, leakage of blood into the surroundings, these areas, and then the person's vision is usually affected. So a person with normal vision may see something like this. A person with diabetic retinopathy may have problems like this, blurred vision like that. There are also many other aspects in terms of what it does. One of which is it can have renal diseases, because the kidney is filtering, is kind of overworking. One thing is having high glucose is one of the problems, and kidney is constantly purifying the blood and removing the glucose as much as possible, and that is one of the problems that is going to happen. And cardiovascular disease, that is a 2 to 4 fold increase in cardiovascular diseases. And extremity amputations, that is another issue that we will discuss in the next slides. So if we see the extremities, like mostly that is of the feet, a healthy feet will have constant, two things are required. One is the constant blood flow and nervous stimulation from the nervous system. That is, the nerve should reach till the end. In cases of diabetes, the person, the vascular, the lumen, or the blood vessels narrow down, and the nerves are damaged. So because of which the leg, the feet may not be receiving as much of nutrition, oxygen required. And as a result, sometimes what may happen is there is no blood supply, because of which this tissue will start dying and undergo necrosis, right? What it would look like is some things like this, where because feet is where we are walking on, the whole body pressure is on the feet, and they are likely to get damaged like that. And in extreme cases, they may also happen suddenly, blood supply is not there, and then the tissue is dying out, and there is no option but to amputate the tissue, amputate the leg. So once something has become necrotic, the doctor, the surgeon, has to amputate until where the blood supply is good, right? Why is that important? Is if we are making a cut here and there is no blood supply, how will healing happen? For repair to occur, we still need a lot of blood and other things, immune system of the body. And there should be a good supply. So the doctor would amputate until where there is good supply of blood and oxygen. So there are two things. First thing, we were saying that in the eyes there is hemorrhage, and something like aneurysms may happen. Whereas in the legs, we are saying that the blood vessels become narrowed, and because of which there will be problems. So the angiogenic status in diabetes is kind of twofold. In some tissues, it is anti-angiogenic. That means blood vessels are not formed enough, like in the heart, kidney, wound repairs, blood vessels, and so on. In those cases, they are kind of anti-angiogenic. I hope you have seen there are a lot of there is a market for diabetic footwear, right? They come with full covering and very soft soles and other things, so that they can avoid getting damage. Okay, so in some tissues there is anti-angiogenic, and in some tissues they are pro-angiogenic, like in the retina, kidney glomerulus, and cancer. In those cases, the angiogenesis is much more than otherwise normal. So what to do is the question. As we discussed, type 1 and type 2. Type 1 is the lack of insulin, so we should supply insulin. Type 2 is because of insulin resistance, and we don't have proper treatments for it. So we can manage diabetes to an extent, that is, changes in the diet and proper workout, exercise should be there, and then some medical supplement. And if it is diabetes 1, we should supply with insulin, right? For diabetes 2, there is no point of giving more insulin, because it's the person is already, the cells are, it is about insulin resistance. So one of the things we should try to remember, if you excuse me 1 second. Okay, sorry. Anyway, I wanted you to also, because you're young, and how you lead your life during the young age is more important to have to age well over the during the later part of life. So you have to remain active, and there's the principles are simple. It's hunt, eat, sleep, right? This is what every animal does, and that is what we are supposed to do. And we have dysregulation in these three activities. First thing is we are hunt. Here what I meant is working for food. We are no longer working for food. There is no physical activity. There are, I think in the last class too, I said about, I discussed something like this. There is, if this is the whole day, say 7 a.m. to 8 p.m., say for example, if that is your whole day, we spend, maybe in this time we may have work and exercise around say 4 to 6 or 4 to 7, and the rest of the day we are remaining sedentary, sitting or sleeping mostly. The animals usually don't do it the other way. They keep working. Their work is spread throughout, and even research has found out that those people who are having activity, even small amounts spread throughout the day, are much healthier than those that don't work at all, or those that just train for a certain period of time, one period of a day. So spreading out physical activity, the problem is not about working or not. The problem is being sedimentary, being sedimentary for too long. Yeah. Okay. So hunt is about work. Physical activity should have been there. That is one problem. Second one is eating. We eat more than we require, and we eat just because we are bored. We eat just because it is there. It's something like that. Our eating is disproportionate, highly disproportionate to our activity. And then the other one is sleeping. You can look at monkeys or other, not the domesticated animals, but still those that are continuing their lifestyle. Maybe they sleep by 6 p.m. or 7 p.m. They wake up by 5 a.m. or so. That is how we are all biologically, actually biologically designed, right? We are not nocturnals. If you compare it ourselves with chimps and other things, typically this is our routine somewhere. But we push it to 10, 11, 12 a.m. and wake up late. The point, this is sleep is not so much of a matter apart from the dysregulated circadian rhythms people may be having, because circadian rhythms is also related to metabolism. So sleeping early is good, and waking up early is, sleep early, early to bed and early to rise. That's the best thing that we have. So based on our lifestyle, even the job that we do, we are highly prone to these lifestyle diseases, especially the something like diabetes mellitus type 2. So um, what do we have for reducing the blood glucose. Blood glucose is the problem that is being caused, that is type 2 diabetes. In that case, it's not because of insulin, lack of insulin, it is because of insulin resistance or resistance to insulin. So there are several drugs which reduce the which reduce the increase in blood glucose, right? How does blood come from? It comes from the intestine by absorption. Here we reduce the absorption so that blood glucose doesn't increase. We can also increase, or sorry, reduce the glucose release from the liver, which is a storage of in the form of glycogen in the liver. That is another way. And we can try to increase the glucose uptake by the muscle cells, right? And also increasing the insulin release as well. So the target is somehow to reduce the blood glucose. So release of blood glucose into the blood, or increasing the uptake by other things. Here, these are all drugs. Okay. If every drug that has an effect is likely to also have side effect. And if there is no effect, I doubt if there is no side effect, I doubt if it is effective at all, right? We need to think on those lines as well. Anything we put in abnormal into our body, and we cannot claim that it is going to be very good only, right? It is going to have some effects, and we need to minimize those effects. That's what we try to do. So what is the ideal therapy? So the ideal therapy is to replace the damaged tissue, right? If we, at least in part for diabetes type 1. Type 2 is a complicated disease because we have to, it's not about insulin, lack of insulin, but it is about resistance to insulin. So it is a complex one to treat. So there are 2 points, and I wouldn't spend much time. At the same time, when we speak about hyperglycemia, lower blood glucose also can have effects. If you are taking medicine, or if you are taking any insulin, for example, and that might reduce the hypoglycemia, cause hypoglycemia, which has its own problems, right? So we should be, any ideal thing we are thinking of, we should not, there should be a balance between these two. That should be in the normal range and not go very high or go very low. First, let's see what is insulin. Insulin is a very small protein, actually. It has about 21 amino acid chain, 2 chains of 51 amino acids, that is chain A and chain B. And there are 2 disulfide bonds between these 2 chains. So it's a kind of simplest protein with a quaternary structure, you can assume that. Of course, it's not produced as it is single polypeptide produced and then becomes 2 chains. We'll see the processing of it later. So people, through recombinant DNA technology, we are now in a state where we can produce both these chains, chain A and chain B, like this, and chemically make them add disulfide bonds and give it to the patient. In the previous days, I think they used to use pig insulin. So they would have gotten a lot of pancreas from the slaughterhouses of pigs, and then they would have taken isolated insulin from there and then give it to the patient. That was in the very olden days, beginning of the time when people were trying to treat it. But imagine, so many pigs had to be slaughtered, or basically they would have used for pork and meat industry. But from so many pancreas, we would only get very little of it. But now we have recombinant DNA technology. We can make E. coli produce the alpha chain, sorry, chain A and chain B, and then we can make this, and then we can give it to the patient in large quantities. Majority of it that we have it is recombinant insulin. Sorry. There are still, if we want to produce insulin or make cells, excuse me one second. So there are several challenges before we try to address the problem. Before we try to provide a solution, we should have understood the problem well. First thing is, if we are trying to provide a cell-based treatment, right, like the stem cell one, there should first be there should be insulin gene expression. Insulin gene is present in all the cells, but only the beta cells produce insulin. So there should first be insulin gene expression. Second thing is, insulin produced should be processed. Insulin is actually, I think I must be having a slide somewhere, is produced as single polypeptide. Then it folds like this, disulfide bonds are made, and this part of it is degraded. That's how we get chain A and chain B, and chain C is deleted out. So we need to have all the proteases required for processing of that insulin, proinsulin. Then when should insulin be um, when should insulin be produced? Insulin should be produced, or when should released? When should insulin released? It should be released only when there is high blood glucose, usually after a meal. How much should it produce is also important. And then when the blood glucose has reduced, it should shut down the insulin secretion as well. And the cells that we are putting in, they should be for a long duration. It's not like every year you have to come and get cell transplantation, beta cell transplantation. That's not an ideal solution. And then these cells, I mean, longevity and survival both are in the same thing. They should be together for a long time. And the cells that we are putting into the body should not interfere with other cells, because we have alpha cells and delta cells and other cells within the region which should not be affected. Okay, let's see a little bit about the production of insulin. Insulin is produced as a single polypeptide. It's called as pre-pro-insulin, which is having a signal sequence for during translation, which is important. And that part will be cut out. And now we have pro-insulin. That pro-insulin will be further broken down. And then we get the final insulin. Okay. That is where we have the chain A and chain B. So during the signal peptide is important for translocation into endoplasmic reticulum, where it is, and in the Golgi complex it is processed, and then it will form a exosome into which you have the insulin produced. Upon signal, these exosomes will be, or not, the vesicles will fuse with the membrane, releasing the insulin out into the bloodstream. This is how sensing happens in a normal beta cell. When glucose is taken up, right? Glucose is produced. Right after meal, blood glucose increases. And beta cells have something called as GLUT2, glucose transporter 2, which is facilitated transport, which takes up the glucose. The glucose that is taken up is phosphorylated by glucokinase, and that will further enter into glucose 6 phosphate, goes into glycolysis. Then there is increase in ATP by ADP ratio, that is the energy source. The increased ATP will bind to the potassium, ATP sensitive potassium channel. When it binds, it blocks it, and then there is membrane depolarization because of you have high potassium ions within the in the cytoplasm. That triggers another channel, that is voltage gated calcium channel. Calcium is always, or majority of the time, is away from cytoplasm. So this is nucleus, this is the extracellular side. Calcium is stored in the smooth endoplasmic reticulum or in the extracellular side. That means calcium is not typically present in the cytoplasm, right? But upon stimulation, the voltage gated calcium channel opens up and extracellular calcium ions flow in. And these calcium ions will, will allow the vesicles containing insulin here to fuse with the membrane, releasing the insulin out. I hope you understood the sensing mechanism that beta cells have. It's a fantastic way of doing stuff. Every time glucose is there, GLUT2, ATP ADP ratio, then potassium channel, ATP sensitive potassium channel, then voltage gated calcium channel, and then insulin vesicles fusing with the cell membrane and releasing insulin out. What happens with the normal cells then? What does insulin do? Glucose, the insulin, this is insulin receptor, that is where insulin would have bound. If insulin is bound, it will activate insulin substrate 1. This is from carp. I hope you have read it in your cell biology, which will stimulate PI3 kinase, that will stimulate PDK1, PKB, and then PKB will mediate something else, that is GLUT4 containing vesicles. These are transporters that are present as vesicles in the cytoplasm. Because of the activity of PKB, the vesicle is allowed to fuse with the cell membrane. And now these glucose transporters are exhibited on the cell surface, and then the glucose will be taken in, right? Glucose is transported by um, facilitative transportation. It's a kind of passive one. You have 10 glucose molecules and one here. There is a gradient because of which, say, 0 we will take. So 5 will have come here. These 5 glucose would have been converted to glucose phosphate. And how many glucose are there? 0. So 2 more or 3 more will come in here. And that is how the glucose is taken up from the blood into the cytoplasm. And then that glucose can be converted into glycogen or used for the purpose. When there is excess glucose, it will be transported and it will undergo glycogen synthesis. Okay, in the beta cells, we had GLUT2, and in the insulin responsive cells, we have GLUT4 transporters. Okay, this is how it is summarized. If you can see, this is a beta cell, and this is a muscle cell, skeletal muscle cells you can think of. So where do we start? This is glucose. GLUT2 takes up, ATP increases, ATP sends to potassium channel, deep membrane depolarization, voltage gated, voltage dependent calcium channel opens up, calcium ions are released or enter into the cytoplasm, because of which insulin containing granules bind to the cell membrane, releasing insulin into the blood. That binds to insulin receptor, which will activate multiple things, including the one for glucose uptake here. Okay, and there are also other things where you have cell growth, increase in metabolism, and also glycogenesis as well. So what we have learned so far is the biology of how these things happen. Without that, we don't know what we are approaching. We should be able to, when we make a choice of how we want to do what we want to do, we should have known what the problem is. I hope you know what is meant by first principles. please note it down and try to watch some YouTube video on that. Just breaking down the problem into basic components and then trying to address it. That is exactly what we would also try. But here we are doing it theoretically. Okay, if insulin production is the problem, then we need to have some beta cell replacement. One approach is allotransplantation, that is, you take beta cells from somewhere and then put it into the individual. That could work. The other one is beta cell regeneration. Maybe there are some reservoir cells, and somehow we stimulate those cells to divide and become beta cells. That is one another approach. Um, there are similarly xenogenic islets, and there is one more approach is immunoisolation. We will see what it means, of course. In the first place, if it is diabetes 1, type 1, we are saying that the beta cells are killed, right? And what is the guarantee that if we are going to put beta cells, why wouldn't they be killed too? Right? That is also, we have to think about that as well. We might be able to get the beta cells perfectly fine and all, but as soon as we put, or within 10 days, if those cells are killed by the autoimmune responses, for example, then everything is futile. So we need to think about that as well. Here are a few examples about approaches. We are not, I don't need you to mug up all the names of these genes, but rather I would want you to think about how you would, if you have understood the problems of the what is the biological problem with the diabetes and the functioning of a normal tissue, and you're trying to imitate the normal tissue, and then how would you do that is the question. From stem cell biology perspective, we have several approaches. We could have taken embryonic stem cells. We could have worked with induced pluripotent stem cells. We could have taken umbilical cord cells, or we can take bone marrow. So blastocyst, from which you would have taken embryonic stem cells and then allowed it to grow into an embryoid body, and then taken the endoderm, and from there we can have using using components like these, we can probably differentiate these endoderm cells into insulin expressing cells, and those cells could be given. Similarly, induced pluripotent stem cells. In genetic reprogramming, we have learned a little bit that OSKM or OKSM factors could be overexpressed using genetic methods, which of course have a risk. And the fibroblasts could be reprogrammed into induced pluripotent stem cells. That is, they are almost equivalent to embryonic stem cells, and then grow them with certain factors because of which they could differentiate into insulin expressing cells. So this is possible with umbilical cord blood cells as well. The bone marrow is a good source of embryonic stem cells because when we take the bone marrow, there are abundant mesenchymal stem cells. Of course, it's not the easiest one, but still, considering many other situations, this is a kind of good source for bone marrow. We could have used the same person's bone marrow instead of taking, say, this should have come from somebody else, right? Embryonic stem cells. Cord blood is coming from somebody else almost. And fibroblasts maybe from his cells, and we are doing induced pluripotent stem cells, but these are usually not um, identical to the embryonic stem cells, in the sense there could be some issues with gene expression. In that sense, if we see, bone marrow is a good source of the autologous source, okay, and then take mesenchymal stem cells and then we can probably make them express into beta cells. Okay, these are all clinical trials. You can find many papers making very little bit of progress towards solving it, and we need to understand and study more because there are so many people having issues with diabetes. One of the best solution, I think, is trying to have insulin producing cells from progenitor cells. In all these cases, the previous ones that I've discussed, is we take a cell type, one cell type, and we are trying to make it into a cell, some beta cells or insulin producing, we'll just say it as beta cell. In embryos, each of these cells would have grown into specific lineage, right? Endoderm, mesoderm, and ectoderm. And they would have their own paths or differentiation lineages like these. When we say lineage and differentiation, change in form and function, these cells in each step are having variations in gene expression. How can we just take one cell type and directly make it into beta cell and assume that their genetic or nuclear program, right? The epigenetic nuclear program is identical to that of the naturally occurring beta cells. There could be problems. So, what to me, why I feel I think I would go with this is trying to use some progenitor cells which have already, say if it is coming from the endoderm, it is already here in this stage, and a little more it might be forming a pancreas or say beta cells. If until here at least the gene expression or the nuclear program is very similar to that of beta cells, it has to take maybe a few more steps to become a beta cell, right? The differentiation would be easier and uh, as authentic as possible, right? If we take a cell from here and directly go to beta cell, we cannot be sure that all the nuclear program is accurate. When I say nuclear program, it is which genes are turned off, which genes are turned on, and how much are they turned on, and many other aspects of gene regulation and gene silencing too. Yeah. So in that sense, I would like the progenitor cells, especially those of the liver and pancreas. They are coming from the same tissue or same progenitor cells before, and then eventually set will become liver and one set will become pancreas and then beta cells. That means their nuclear program is highly related, and it could be easier for one to become the other one. So we can probably take those, say you can try the liver, we can try pre-existing, if there are any pre-existing beta cells, we can probably take them and try to multiply them. That is another way of doing things. We can also take the other stem cells that are present in the pancreas and try to differentiate using some of these factors and hopefully generate insulin expressing cells. Okay, you don't have to mug up all these names, that will be just the memory based, but I want your thinking to be right as if it is a troubleshooting. You have a problem, or if I gave you several strategies, which one do you think is better and why is it better? And the points that I'm talking about are the precautions or the approach you have to keep in mind when you're troubleshooting, right? The trouble is diabetes and you're trying to provide a solution, and how would you do that? There are several problems and challenges that are associated with, like every cell, every treatment, we also have problems. First problem is, we say diabetic person, if it is type 1, they're dead or dysfunctional, and they could be dead because of the antibodies against beta cells. In that case, if we have derived beta cells and put in the person, they could have been killed. So what should we do is, one of the solutions people propose is to put it in a immunoprotective device so that the antibodies don't reach and kill the cell, and then put it there, and these cells keep on producing insulin when and where required. Please remember that beta cell is the best replacement for a beta cell. Why is that? The timing, amount of production of insulin, everything is served best by a beta cell. There are many other sources as well. People have tried to produce, and they've tried to do it in with mice. There has been considerable success too as well. But in my opinion, beta cell would be the best of the solutions. But the problem is we have antibodies against it, and that's a danger. So again, putting in a immunoprotective, any device like this could cause biofilm problems or infection issues. And that means that is another problem we will have, right? And yeah, but otherwise we need to keep into the how long will the implant be there. And some people, there are also non cell based types, like putting insulin patches, injections, and many other things, but the timing is not very accurate, or the amount is not accurate, and there is always need for supervision. When a person has to do it on a daily basis, then it becomes a little bit troublesome. Okay, these are some of the questions you might want to look at. But otherwise, I'll stop here. If you have any questions, you can ask. Anybody?